Did you know that trans fats are considered unhealthy for you? Look on a nutrition label. Many times you will see trans fats. If you do see these a lot, and there's a lot in that food, that's probably not good for you. The word trans in trans fats is related to the structure. And so the idea that organic chemistry has this topic called cis-trans isomers tries to help us understand structurally the difference between a cis molecule and a trans molecule. Fatty acids are molecules, so when you talk about trans fats, you're talking about a structure that has a trans isomer versus a molecule that might be a cis fatty acid, which are considered healthy, and cis fatty acids have a structure that's in a cis conformation. And so this topic we are interested in because, well, cis-trans isomers are important. Our bodies can tell the difference between a cis isomer and a trans isomer, even though our eyes might fool us if we looked at the structure. Look down here at the bottom. These two structures, one of them is in cis and one of them is trans. Both of them, if you tried to name them just using organic nomenclature, would be, you might say they're both 2-butene, and you would be right. But one of them is cis-2-butene, and the other one is trans-2-butene. And it turns out this one is the cis, and this one is the trans. And so we need to talk about how, when looking at structures, we can tell the difference between a cis version of a cis isomer structurally versus a trans isomer structurally. If you hang on to the end of the video, I will give you another example for you to see the importance of cis-trans isomers in human health. Now, this video is the second in a series of videos on isomers, so if you want to see the first video or you want to go deeper into isomers, you can watch some of the later videos discussing different types of isomers. Now, all isomers, one of the conditions of all isomers is that the molecules that you're comparing must have the same molecular formula. This was true in structural isomers that I've already have a previous video on, and it's also true with cis-trans isomers. For you to have cis-trans isomers, the second thing that must occur is that they must have the same connectivity of atoms. In structural isomers, again, previous video, the connectivity of atoms would be different between the isomers. So this is where structural isomers differ from cis-trans isomers. With cis-trans isomers, they have a different spatial arrangement of atoms. And so the next few slides we'll talk about that spatial arrangement and what that really means. First important note to know about cis-trans isomers is that not any molecule can have a cis-trans isomer. Only some of them can. In fact, there's about 13, 14 families of organic molecules, just as typically what is covered in an organic chemistry course. But only 2 of them can really have cis-trans isomers, and that is the alkenes. And alkenes are the ones that have the carbon-carbon double bond in them. And the other one is the cycloalkanes. So as an example, I can put this cyclopentane molecule here. Those are the 2 families that really have cis-trans isomers. So if you're given a question and then they ask about isomers and it's an alcohol and they ask, does this have a cis-trans isomer? The answer is no. You really don't have to evaluate it. Alcohols can't form cis-trans isomers. They have to be alkenes or cycloalkanes. And most of the time, the one you're going to see in your organic chemistry, at least the one you'll see first, is the alkenes one. Organic chem courses that go deeper into isomers will start to cover alkanes as well. And so these videos are oriented in a way that I kind of go from simple isomers to more complex. And so therefore you can kind of stop watching these videos when you get to the point where you think it got to the depth of your course or the depth of the knowledge that you want to know them by. So in this video I'm not going to cover cycloalkanes because typically, again, alkenes are the families that you'll actually be associating with cis-trans isomers more. So let's just focus on cis-trans isomers of alkenes for this video. So of course alkenes are hydrocarbons that have a carbon-carbon double bond in them. And so here you see the carbon-carbon double bond, and we know in structures that carbons like 4 bonds, and so each of these carbons are going to be double bonded to the other carbon, but also have 2 bonds that connect to something else. These are R groups. These are some sort of hydrogen or carbon-hydrogen group hanging on them when we're talking about an alkene. And so for our discussion, I'm going to put some variables here or letters here just to differentiate the 4 different spots, or the 4 different hydrogens or hydrocarbons that could be hanging on there. Like maybe A is a hydrogen and B is a methyl group, and D is an ethyl group, and E is a hydrogen, that kind of thing. So those are just the 4 things hanging on it. But no matter what those 4 things are, this is an alkene because there's a carbon-carbon double bond sitting there. So I've drawn here 4 structures down below. And what we're going to do is kind of figure out what is the relationship between each of them in terms of being isomers of each other, and maybe for this video, are they cis-trans isomers of each other? So the first thing is we have to make sure they have the same molecular formula. And when we look at the molecular formula of any of them, we see that they are C4, 3, 4, 5, 6, 7, H8, I think is what I got. C4H8. So if you look at all 4 of those, you'll see that they each have the same molecular formula, which means we can now go on to the second test of isomers, because remember, they have to have the same molecular formula to even have a chance to be isomers. So these have a chance to be isomers. Let's look a little bit deeper as to what kind of isomers they might be. So the next thing we want to do is see if the arrangement or the arrangement of the atoms are consistent. I'm sorry, the connectivity of atoms is the same between them all. And so to do that, we can go through carbon by carbon and kind of determine how they're organized. Well, the other way I like to do is just let's name them. And so if we go to name them, I'm not going to spend time naming these guys and going through the process of naming them, but the 2 molecules in this column are 2-butene. The ones in this column are 1-butene. If the names, if they have the same molecular formula, but the names are different, as they are here from our normal organic chemistry naming rules, like they are here, without worrying about isomers just yet, but just naming them, if the names are different, it means the connectivities are different. And so what that means is the 2-butene molecules and the 1-butene molecules are not cis-trans isomers. They are not cis-trans isomers because the names are different, but the molecular formulas are the same. What these actually are structural isomers of each other. And so that's where the structural isomers from the previous video we talked about, that's what they would be. So for instance, if you look at this molecule, a 2-butene molecule, and compare it to this molecule, a 1-butene molecule, and again, I could have picked any 2-butene molecule and any 1-butene molecule, the answer is the same. If I compare these two to each other, those are not cis-trans isomers, those are actually structural isomers. They have different names, same molecular formula. Now, when you're naming alkenes, a light bulb should always go off in your head, or an alarm should go off in your head saying, when I have an alkene, I have to think about cis-trans isomers. When I name it the way I normally name a molecule and I think I'm done, now we have to take one more step and think, what about cis-trans isomers? If you're naming an alkyne, if you're naming a thiol or an ether or an alcohol or an aldehyde, no, you don't need that alarm. But these alkenes, they have this cis-trans thing that you always got to consider. So you go name the molecule, and then you evaluate, does it have cis-trans isomers? And that's what we're going to do now. We already named them. We named them 2-butene and 1-butene. Let's take a look at the 2-butene molecules, these two, and let's compare those two. They have the same molecular formula. They have the same connectivity of atoms, i.e. they have the same name we would give it. Now we just have to evaluate, are they actually cis-trans isomers? Now, cis-trans isomers are one of the more difficult things, I think, to really and conceptually understand structurally. So it takes a moment and takes some time to really appreciate this. But there's a method you can go through to determine whether one is cis and one is trans. So let's do that. So just because it's an alkene doesn't mean it has to have a cis-trans isomer. So when we evaluate for cis-trans isomers, we have to first ask ourselves, can it even have cis-trans isomers? And so that's the first part of the evaluation that we're going to do. And to do that, we're going to look at the two carbons involved in the carbon-carbon double bond. So we'll be looking at what's attached to them. So for that, let's go back up to our little model up here so we can use this communication vocabulary. So notice to one carbon, group A and group B are attached. To the other carbon, group D and group E is attached. Let's look at the first molecule here in our 2-butene column, and you can see here that if we match it up, A is here, it's this methyl group, B is this hydrogen, D is this methyl group, and E is this hydrogen here. Now, I just removed all those marks so that otherwise this is going to become very messy. But what we do now is we compare what's attached to one of the carbons of the double bond. In this case, well, in our first example here, we're going to compare group A and group B that's attached to that first carbon there of the double bond. And we ask, are they the same? Is A and B the identical group hanging on there? Are they exactly the same? And you can see that A is a methyl group, CH3, and B is a hydrogen. Those aren't the same. If they were both hydrogens or both methyl groups, oh, that's the same. These are not the same. Okay, so they are different. Now let's look at D and E. And you can see when we look at D and E, one's a methyl group, D, and E is a hydrogen. Once again, D and E are not the same. If both carbons, if A and B are different, and D and E are different, that molecule has cis-trans isomers. That's the test to determine whether the molecule will have cis-trans isomers or not. And so this molecule here has cis-trans isomers. So 2-butene has a cis isomer and 2-butene has a trans isomer. And so now what we want to be able to do is figure out which one is it that we're staring at. And to do that, all you do is you run, or I like to run, a dotted line through the carbon-carbon double bond to split the molecule in half. You can think of that dotted line as railroad tracks where a train would run through. And you ask yourself, who lives on each side of the train tracks? So again, you draw the dotted line through the double bond and you have two molecules or two groups hanging on each side. On one side of the tracks, I have the CH3 and a CH3. On the other side, I have a H and an H of the railroad tracks. If the H's are on the same side, that is cis. If the H's are on opposite sides of the track, that is trans. So this molecule right here is cis, and cis is CIS, is cis-2-butene. Now let's look at the bottom molecule in the 2-butene column. So let's look at this one. So again, I've named it as 2-butene, but the question is, because it's an alkene, I need to make sure that I have to check to see if it has cis-trans isomer possibilities. So again, what we do is we compare A and B, we compare methyl with hydrogen, and we see that they are different. We move on. We move to the other carbon and we look at D, which is the hydrogen, and E, which is a methyl group. Again, different. If A and B are different and D and E are different, both groups have to be different, just as they are here. A and B are different from each other and D and E are different from each other. This molecule has cis-trans isomers. And so what I do is I drive the train tracks through the double bond, and notice I have a hydrogen on one side of the tracks and another hydrogen on the other side of the tracks. So they live on opposite sides of the tracks. If they're on opposite sides, this is trans. And so the bottom molecule is trans 2-butene, would actually be its full name, and the top molecule is cis 2-butene. Now let's look at 1-butene molecules that are shown in the right column there of the two structures below the 1-butene verbiage, and I just boxed them in right there. So those are the ones we're going to look at now. So looking at those two molecules, or let's look at the top one first, and let's go through and determine whether these are cis-trans isomers. So again, these are alkenes, so our little alarm goes off saying, hey, I've named it as 1-butene, but I better check to see if there's cis-trans possibilities here. And so what we're going to do is we're going to compare A and B first and see if they are different. And when I compare A, I circled that one, and B, notice they are the same. They're both hydrogens. If they're the same, there is no cis-trans isomers possible with that molecule. So there are no cis-trans molecules. So 1-butene would be the proper name. There's no such thing as cis-1-butene and trans-1-butene. Notice I know D and E are different, but both A and B must be different, as well as D and E. In this example, D and E are different, but A and B is the same. If one group is the same, there's no cis-trans isomers. So there's no such thing as cis-1-butene or trans-1-butene. If we look at the molecule below, we have the same problem in that D and E are identical, therefore no cis-trans isomers with that molecule. So in fact, these two molecules are the same. These two molecules are different. The different ones, the two I just did and looked at, the 2-butene molecules are different. They are different because they are cis-trans isomers. The 1-butene molecules, the ones that are the same on the right-hand side of this slide, those two molecules are the same molecule, so you are actually drawing the same thing twice. Now you may be wondering, why can't this molecule and this molecule, you just rotate it and you move this CH3 down to here and the hydrogen up to here by rotating the molecule to get those to interconvert, so really they're the same molecule. You might be wondering why that is. And so now let's get into the reason why these isomers exist in this way. And that is, let's just say we took this molecule. A carbon-carbon, whoops, that's a single bond there. And we have a CH3 and a hydrogen and a CH3 and a hydrogen. Okay, I know first of all, that molecule is completely wrong because those carbons in there don't have 4 bonds to them. But let's just say they have a single bond there between them, and let's just say this is a real molecule, just for argument's sake for a moment. Because there's a carbon-carbon single bond there, a carbon-carbon single bond can rotate freely. And so that it can rotate freely means that we could just twist this carbon right here, and when we twist that carbon and turn it, and it rotates on this bond right here, this CH3 will move down to here, and this hydrogen will move up to here. Kind of like turning a crank, turning a wing nut, or turning a wind-up toy. You're kind of doing that thing. And when you turn that wind-up toy, you can see that the two edges would flip positions. And that's all fine and great. And carbon-carbon single bonds can do that, which is one of the reasons why they don't have cis-trans isomers. In carbon-carbon double bonds, this bond right here, that double bond, there's no rotation around that. It cannot rotate. We can flip the whole molecule. You can flip it upside down and flip it left to right and all that kind of stuff, but you cannot rotate it on that carbon-carbon double bond. Because of that, we can't interconvert these two molecules. Those cis-trans can't go back and forth. The only way that we could do that is if we broke the bond and made it a single bond. And if we broke the bond and made it a single bond, then we could rotate it there. But the problem is, to get that bond to break takes a lot of energy. It takes enzymes to do that. And so there are chemical reactions that can convert cis isomers to trans isomers. That can occur, but if the molecule is just sitting inside of a cell, inside of you, it just doesn't convert from one to the other on its own naturally. Where an alkane with a bunch of single carbon-carbon single bonds, those can rotate freely sitting in the cell with no help. They'll freely do that. Much like you can raise your arm up in the air and lift your leg, and you can change your positions very easily. Carbon-carbon single bonds rotate very easily, so their shapes change a little bit more. When you have a carbon-carbon double bond, that doesn't rotate there, and it's locked into that position. Now notice these two molecules here, these 1-butene molecules, those two are the same molecule. And you might be wondering how that is. They don't look like the same molecule. They're kind of arranged a little bit differently. Visually to my eyes it looks a little bit different. But think of these molecules as pancakes for just a moment. We're going to flip it. We're going to flip the pancake over. And so kind of like we're going to turn the page. So if this is the binding on the page, let me change that color. I don't like that color for this. Let's take the top molecule. There's the binding on the page, and we're going to turn the page this way. And when we do so, this molecule, this pancake, is going to flip upside down because we're turning the page over the binding here. And when you flip that over, what does that molecule look like? It's going to look like this, where this part comes way over here, and this hydrogen is a much closer to the edge of the page, i.e. this one and this one. And so when you flip it over, because again we don't rotate around those carbon-carbon double bonds, when you flip it over like a pancake, it looks just like this molecule down here, i.e. they're the same. Now you might be thinking to yourself, well, the carbon would be written backwards if I flipped it. Yes, but that carbon is not really what that atom looks like. Think of the atoms as a bunch of spheres. Whether you flip a sphere upside down or not, if you flip a basketball upside down or not, it still looks exactly the same and acts exactly the same. So when we flip that molecule over, we're not breaking any bonds. We're just taking the whole molecule and turning it upside down. Notice if we do the same thing with this molecule, the 2-butene molecules, take the top molecule here, and we're going to flip it. We're going to flip the page over that blue line, so we're going to flip it like a pancake. When you do that, it doesn't look like the molecule at the bottom, right? Notice when you flip that over, the 2 methyl groups are going to be down at the bottom, and the 2 hydrogen groups are going to be up a little bit higher, i.e. it'll still be in cis conformation, whereas this one at the bottom is in trans conformation. And so if you flip that molecule at the top over that blue line like a page or like a pancake, you're going to flip it over. Now the 2 methyl groups and the 2 hydrogen groups are still on opposite sides of the train tracks. It's still in cis, and it doesn't match that molecule at the bottom. Now I'm going to post another video with practice problems of naming alkenes with the cis-trans designation. So if you want to now put this into a little bit more practice, I'll have the video for you to go through. So it'll be just a bunch of practice problems, not explaining how to, and I will explain how to name them as well. So I'll actually walk you through it as well if you're still struggling a little bit, or maybe to confirm that you're getting it, which I sure you are. The last thing I mentioned I was going to talk about was one more example of cis-trans isomers in human health, and that is our vision. Our vision is controlled in part by isomers of an alkene. And what happens is when light comes into our eyes and hits our retina, there's a molecule in our retina called 11-retinal, and it exists in the cis form and the trans form. And when light hits our retinal molecule in the back of our eye, it changes from cis to trans, which activates our vision processes within the rod cells of our retina. And so here again, this act of light hitting the back of our eye for us to be able to see, it occurs because you have a molecule that converts from cis to trans. And notice for that to happen, it has to break the double bond of 11 cis retinal. So it breaks that double bond. When the light hits it, it gives energy to that molecule. It breaks that double bond. The molecule rotates around the single carbon bond and then reforms the double bond in the trans conformation. That whole act stimulates the activity in those cells, which then signals to our brain that there is light there, that there's something out there, that we're seeing something, that we're seeing some vision. And so this conversion from cis to trans occurs in our eyes as a very important role for us to see things. But when it comes down to it, it's basically just an alkene. The double bond is being broken. That's in the cis conformation. It rotates around that single bond because it doesn't have the double bond anymore, reforms the double bond in the trans conformation, signaling the events in our brain for us to be able to see. Pretty cool. Thanks for listening.